Okay, great. I think we are ready to begin. Yeah, it's 9 o'clock. Good morning, everyone. It's really nice to be here. I want to thank, first of all, the organizers of OHBM for this wonderful conference. Welcome my fellow speakers and all of you to this morning's session on PET imaging. Now, our aim for the next hour or so is to show our work, our studies that we do combining PET and fMRI. And our aim is really to show that the addition of PET imaging to studies of fMRI activation and connectivity can really help understand and supplement our understanding of fMRI activation and connectivity. And I think that is particularly relevant as the field of human brain mapping moves towards a more integrative view and models of brain function that also considers metabolic and molecular processes that shape connectivity as we measure it with fMRI. We have prepared 4 presentations for you. So Alireza Salami from Sweden, Karolinska Institute and Umeå University will begin with a talk on molecular and functional pathways of cognitive decline in aging. And then we have Claire Ciampa from Brandeis University. She's a PhD student with Anne Brandeis, and she will talk about dopaminergic enhancement of hippocampal processes in aging. Then I will give a talk myself on age-related changes in working memory and what PET can contribute to that. And then finally, unfortunately only via video, we have Sucru Barres Demiral from the NIA, National Institute on Alcohol Abuse and Alcoholism in the US. And he will just showcase a bunch of studies that they have done in their lab. We hope that we can present a range of different statistical approaches, a range of different designs that we have used, but there's also a common theme across it, and it's not only PET imaging, but it's that we are all particularly interested in neuromodulatory systems, and in particular dopamine. So all of our presentations will feature some form of dopamine receptor PET. And with that, I would like to invite Alireza Salami for the first talk.
Great, thanks, Anna. Good morning, everyone. Special thanks for joining us this morning, despite the club night last night. So special thanks to the organizers to be here. It's a wonderful conference and a great city, so we are really excited to be part of the conference. So as Anna alluded to, in the talk, we're going to talk about different aspects of the different molecular aspects of cognitive aging, with a particular focus on the dopaminergic system. So most of the research that we are doing in the lab, but which overlaps with the common theme of this symposium, is with the ultimate goal of better understanding the neurocorrelates of cognitive decline in aging in order to inform an effective intervention program that ultimately mitigates cognitive decline at the older age. And to pursue that vision, we focus on certain key points, one of which is to acknowledge the huge inter-individual differences that exist across the adult lifespan, but particularly among the older individuals. Second, we examine large-scale longitudinal multimodal imaging data across the adult lifespan, which gives us access to multiple facets of the aging brain. And then finally, we target brain-modifiable factors such as catecholamine system, one of which is dopamine, which could serve as a target for pharmacological intervention. So during this talk, I will first focus on one neurocorrelates of cognitive decline in aging, and that's functional brain organization. I try to focus on characterizing how that breaks down in aging and what are the consequences for neurocognitive decline or cognitive decline. Then I make a transition to the dopaminergic system, different part of the system, pre and postsynaptic marker, try to show how they basically changes with age and what are the consequences for cognition. And then at the end of my talk, I try to link them together specifically to see whether changes in the dopamine system has any consequences for the functional brain organization at different scale. So let's start with the functional brain organization. As we have learned through the past days, it is very well established that brain is a multidimensional manifold with different organizational aspects, and similar to many other biological systems, its organization subserves its function. Assessment of the brain organization, typically through the resting state fMRI, reveal multiple segregated large-scale network ranging from the sensory network to higher-order associative network. And one governing rule of this organization is that the brain regions belonging to the same network are densely interconnected, while the brain regions belonging to the different network are sparsely cross-talking. An important aspect of this is that maintaining this segregated system is very important for optimal brain function and metabolic efficiency. So what about in aging? So with advancing age, we know that older people tend to show less synchronicity within the large-scale network, while they show increased synchronicity between the large-scale network. And these together would result in less segregation of the large-scale brain network or functional blurring. So in a sense, the border between large-scale networks become less clear. So in one study, we were interested to better characterize this, to figure out whether this is more profoundly expressed for a specific brain system, and identify the age at which the system start breaking down in normal aging, and what are the consequences for neurocognitive decline. So using 12-year longitudinal data from the north part of Sweden, we found that particularly the associative system is susceptible to reduction in the segregation in a nonlinear fashion. So we observe a slow decline up to the age of 57, after which there is an accelerated decline. And this is specifically pronounced for the associative system, because for the sensory system, we didn't observe any longitudinal changes, but rather cross-sectional age-related differences. Then we looked into the global cognitive function, which was a composite of several different domains, and we observed a similar nonlinear trajectory, although the decline or the onset of the decline started happening a bit earlier. But most importantly, when we link the changes in the associative system segregation to the global cognitive function, we found that those individuals who decline more in associative system segregation are the ones that decline more in global cognitive function, suggesting that maintaining associative system segregation is very critical for cognitive function. So one might ask, what could be the mechanism underlying age-related decline in network segregation? And to answer that question, I would like to make a transition to the neurotransmitter system with a particular focus on dopamine. So dopamine is an important neurotransmitter which is highly implicated for physiological and behavioral responses. It is largely regulated by at least 3 key mechanisms. One is dopamine synthesis, where dopamine is synthesized in the presynaptic neuron. Then we have dopamine release, where dopamine is released in a synaptic cleft and interact with different postsynaptic marker, the most dominant of which is the D1 and D2 receptor density. And then dopamine reuptake, where dopamine is taken back to the synaptic cleft through the function of the dopaminergic transporter. So in our center, we launched 2 large-scale studies to better understand the the postsynaptic dopamine system with a particular focus on D1 and D2, how they break down in aging, and what are the consequences for cognitive and functional, for cognitive function and functional connectivity. And I mention large scale because these kind of studies may not be comparable to Human Connectome or UK Biobank, but being a PET study, having such a large sample is pretty unique. So in the first study called COBRA, we scanned 180 individuals at the very critical age of 64 to 68, during which the typical cognitive decline starts happening. And we use the reclopride ligand, which gives us access to the striatal D2 system. And we followed these individuals over 10 years. In the second study called Dynamic, we scan a similar number of individuals, but this time across the adult lifespan. And we use a sharing radioligand, which gives us access to the D1 system, which is not only expressed in the subcortical, but also abundantly expressed in the cortical areas. So in both of these two data sets, we have access to extensive MRI sessions, including measures of brain structure and function, as well as extensive age-sensitive cognitive test battery, and genetic and lifestyle factors. So one of the key findings from the dynamic study, which we found support for both cross-sectionally and longitudinally, was the observation of biophasic age-related differences for different brain regions, which was manifested in terms of accelerated decline during early adulthood, followed by deceleration decline during later adulthood. We found several lines of research suggesting that this rate of decline during early adulthood could potentially reflect protracted development, whereas after the age of 40, we observed evidence for degeneration of the dopaminergic system. But most importantly, we found that at the older age, those individuals with lower dopamine D1 system were the ones that were less efficient in the memory function. Using longitudinal COBRA data, we identified two different subgroups that reflected different trajectory. One subgroup reflected a broad-range dopamine D2 decline, which was profoundly expressed in the cortical and subcortical area. And that was the only group that showed extensive cognitive decline, whereas the other group, which didn't show much of the decline except for caudate system, showed preservation over time. And then finally, we also found that the rate of changes in dopamine D2 system was correlated with the rate of cognitive decline across 5-year period. So hopefully these two studies could convince you that dopamine D1 and D2 system are highly implicated for cognitive function. But dopamine is a very dynamic system, and the interaction between pre- and post-synaptic marker might be a better indicator of neurocognitive function. So we recently launched a new study where we're going to scan 140 individuals across the adult lifespan with 3 different tracers to have access to multiple components of the dopaminergic system. One is to use F-DOPA, which is going to give us access to dopamine synthesis. The other one is reclopride, which gives us access to dopamine release during task and dopamine D2 receptor during rest. And then finally using PE2I, which gives us access to dopamine transporter. And here the key question is how the interplay between pre- and post-synaptic marker decline with age, and what are the consequences for neurocognitive decline. And one hypothesis that we have here is that potentially individuals who show more balance in terms of pre- and post-synaptic marker are the ones that preserve their cognitive function across the adult lifespan. So now I will make a transition to the last part of my talk. Hopefully I could have convinced you that dopamine system decrease with advancing age. The border of the large-scale network becomes less clear with increasing age, and the question is whether there is any evidence suggesting that these two are interrelated. Indeed, computational models suggest that dopamine modulate neuronal gain, that is the input-output of the neurons, which enhance the signal-to-noise in neuronal processing and subsequently impact cognition. So we ask a very simple question: does dopamine decrease contribute to alter functional dynamic at different scale? So we wanted to investigate this at the local scale, where dopamine is profoundly expressed in terms of striatocortical organization. Then, given the extensive innervation of dopamine from the midbrain to the striatum and the entire cerebral cortex, we assumed that the loss of dopamine could have a larger impact on the cortical dynamic. And then finally, we investigated whether the organization of the dopaminergic system is related to functional brain organization. In the local level, we mainly focus on the striatocortical organization with a particular focus on caudate, because caudate was the most age-sensitive dopamine-rich region, but there is also a well-recognized functional organization where the central medial part of the caudate is preferentially connected to frontoparietal network, whereas the dorsal wall of the caudate is preferentially connected to the default mode network. So what we observed using both data-driven and quantitative approach was like this distinctiveness in connectivity or organization of striatocortical connectivity was manifested in the younger group, but substantially reduced in the middle-aged and older adult, suggesting that aging is concomitant with the differentiation of striatocortical organization. Then we linked this to individual differences in dopamine D1, and we found that indeed individuals with lower dopamine D1 express less distinctiveness in the functional connectivity. And then finally, we linked the distinctiveness to the offline memory task performed outside the scanner, and we found that those individuals with less distinctiveness of the functional connectivity, less preserved organization, they were less efficient in memory tasks performed outside the scanner. So this would suggest that maintaining dopamine D1 is associated with greater distinctiveness of striatal neural circuits and more efficient memory processing. But as I said, there is an extensive dopaminergic innervation from the midbrain to the entire cerebral cortex. So we wanted to know whether decreases in dopamine D1 system would have a larger impact and impair cortical dynamic. So using resting state data and partial least square, we investigated the joint impact of age and dopamine D1 on a large-scale cortical brain network. And what we found was like indeed increasing age and lower dopamine D1 was concomitant with lower within-network connectivity and higher between-network connectivity. Further analysis revealed that those older individuals with lower dopamine D1 indeed show less segregation of the cortical organization and less efficient working memory processing, suggesting that dopamine D1 signal modulate maintenance of functional network segregation in aging. And then the final part was to investigate the organization of dopamine D1 system. It is very well established that spatial arrangement of the cortical area is not arbitrary, but rather there is a basic sensory motor to transmotor organization, which is apparent in different cortical features in the human cerebral cortex, such as the functional organization that all of us are familiar with. So here we ask a very simple question, whether dopamine D1 express a unimodal transmodal organization across the human cortex, similar to the functional organization of the brain. And looking into the dopamine D1 density as well as co-expression in the cortex, and using a Laplacian manifold learning, we identified a gradient of changes across the cortex from the sensory model to the associative areas with higher density and co-expression in the associative system, which nicely mimic the functional organization that we identified from the same individual during resting state and has also been reported before. So this would suggest that there is an additional dopaminergic layer of the organization. So hopefully I could have convinced you that aging is concomitant with reduced specialization in functional dynamic at different scale. Dopamine D1 signal modulate maintenance of functional network segregation, and also it shares organizational principle with brain function. So at the end, I would like to thank the people in the group who did the actual work, the collaborator, funding resources, and of course, thank you for your attention. I think we decided in favor of time to take questions at the end of the session, so I will move on with the next speaker.